This Week in Parasitism is brought to you by the American Society for Microbiology at microbeworld.org/TWiP. This Week in Parasitism, episode number 62, recorded on October 23rd, 2013. Hi everybody, I'm Vincent Racaniello, and with me is Dickson Despommier.
Hello, Vincent.
You like the...
I do, yes, that's a very nice introduction.
I don't know why I'm doing that.
I don't either.
No, I'm going to do it
It's because I have a new microphone, and we're pretending that we're an FM station somewhere off the coast of Britain, reinventing entertainment as we know it.
Would you like to be live on real
radio?
radio?
That would be great. That would be good. And we could take phone calls and, really have discussions with the investigators.
Well, we can do this. We could do phone calls with this.
We can do that. We can do that.
It's not called phone, but...
Oh, well,
see, to have a radio station, you really need to have a lot of money.
Is that the trouble?
And so we can do it on the internet with no money.
So I keep playing the lottery, but nothing happens.
And we can have people ask their questions online. I just have to set up the tech, and I have.
No, I know. That's the way it is.
Anyway, there is I think a record being set today. I think we're doing 2 weeks in a row, TWiP.
Unbelievable.
Or maybe we did one the week before.
No, I think
it's still close.
We're starting to hone in on the concept of this week in parasitism rather
honing
than this
in on
year
you being here. You're here more often.
Well, that's the reason why we didn't do it every week the last time, because...
Are you going to be here next week?
I hope so.
And you're going to Germany sometime. No, Kitzbühel.
That's in Austria. I will be going to Kitzbühel on November 20th and returning on November 23rd.
That's nothing. That's it.
That's
all your travel for the rest of the year?
I have to give a TEDx talk, which is a privilege to give a TEDx talk up in Middlebury College on November 9th.
Nice.
So I'm driving up and driving back.
It's such a long drive.
To Middlebury, Connecticut? To Middlebury, Vermont?
Yes. 6 hours?
Let's start again. No, it's a wonderful drive. It's the opportunity to listen to all your iTunes, and
you don't
you're
fall
alone
asleep
in the
driving.
car. Well, occasionally, but you wake up alongside. No, I don't fall asleep next thing.
I wouldn't drive that far.
I love driving.
Much.
My sister used to live in Laconia, so I used to drive up and
Was she laconic?
No.
Laconia, laconic?
Yeah, you got it.
Do you know what laconic means?
I do, too. Sort of laid back and kind of disinterested.
The person used very few words.
No, she wasn't laconic.
Should we call this the laconic TWiP? Certainly not the case, is it?
No, not.
TWiP is not
it's
laconic.
Dependent upon the anti-laconic behavior.
You are not laconic, that's for sure.
No, but for long stretches of time, I can be, like yesterday, for instance, which was a nicer day than it is out today.
Today's so cloudy. It's cloudy and crappy.
It's cold. It's starting to get cold.
Cloudy, crappy, and cold. C cubed.
Well, there you go. But yesterday was a gorgeous fall day, so I took it off and I went fishing, which is what I ordinarily do when I can. But I would never go fishing on our TWiP day, never. Or on a TWiV day, I would never do that.
How many more fishing weeks are there left?
Well, it depends on how hardy an individual you are. If you don't mind fishing in the cold, you can fish all the way through December, January.
You put waders on?
Oh, yeah. You got to do that.
Up to your shoulders?
No, they come up to your armpits.
And then you have straps that hold them on?
Yep.
And if you fall, they fill up with water.
I fell yesterday, actually.
And then don't you get, sucked under?
No, you don't. No, you just get wet.
They fill up with water?
Yesterday, I fell right on the bank of the river. And
wow.
It was kind of embarrassing, but it was very
alone?
Slippery. I was all by myself.
You could get hurt and be left there to drown.
I've thought about that. I've actually thought about that. And, you take your chances.
You're not a youngster.
Compared to Galapagos Island turtles, I'm very young. Right. In human years, I'm really young.
As long as you have considered the risks.
Yeah, I have. And they're worth it because, you got to...
Because you could fall and break your hip. Then you'd be there for hours before someone found you.
I'm not in that age group yet, Vince.
Yes, you are.
No, I'm not.
What is that, the 80s?
I'm not in my 80s.
No, the hip breaking group, is that 80s only?
No, it is. Whatever age it is that you start to develop osteoporosis, and I'm not porosis rather. I'm not there yet.
Our colleague next door, who is in his 60s,
It has nothing to do with
is
what I
having
just
his
said.
hip replaced.
I know, but that was an athletic injury that he suffered while playing football at Cornell, of all places. What can I tell you?
All right, let's do some parasitism.
Have we
Just be
covered the
careful.
gamut here?
I just want you to be careful by yourself in the stream in Pennsylvania.
I'm
If there are any
touched.
TWiP listeners in Pennsylvania, whenever Dickson fishes, can you go watch him,
I
please?
wasn't in Pennsylvania yesterday. I was in New York State, but that's okay.
I thought you fish in Pennsylvania.
Well, I do sometimes.
Where'd
but
you go in New York State? Fishkill?
No, I fished on the west branch of the Delaware and on the Willowy Monk rivers.
So I used to go to Cornell as an undergrad,
Yep.
and
Yep.
in my later years I would drive, and I took Route 17, and
Yes.
alongside there's a
Correct.
stream.
That's the one.
It was always full of fishermen.
Yeah, that's right.
and
Depends on when you
waders.
go, but that's true.
Yeah. What river is that?
That's the Beaver Kill.
Okay. You ever fish there?
Lots of times. Yeah, I went there yesterday.
All right.
It was okay.
Today we have a paper and a vaccine.
We do. And
All right.
letters.
And letters. So let's start with the paper.
Sure.
I don't know how I came across. It's probably in my literature scan. It's a paper published in PNAS.
Correct.
It's entitled High Diversity of West African Bat Malaria Parasites and a
It's-
tight link with rodent Plasmodium
Exactly.
taxa.
Exactly.
It's from several groups in Germany.
Correct.
Julian Scher is the first author, followed by Perkins, Descher, Leendertz, Farr, Weber, and Matuszewski.
Correct.
And this was published in its early edition.
Right.
It's,
So it's
somehow at the end of this
recently
year.
out.
Very recent.
September 10th, it says.
A lot of things I learned here.
Yep.
First of all, a bat is a volant mammal.
So it is. And you know what that means, of course.
Flying?
There you go.
Is that a official taxonomic term? Volant
well,
mammal?
it's like the term applied to all flying mammals. Yeah.
So the basis for this paper is that apparently, and you
Yeah.
correct, bats have been known to be hosts for malaria parasites for a long time.
This is true.
I didn't know that,
This is true.
but it hasn't been really studied all that much.
No.
So that's what they did here. They went out and sampled bats and looked for malaria.
Where did they sample those bats, Vincent?
Before we get to that,
Oh, okay.
I want to ask you another question.
Please.
Did you know that?
Were you playing
So-
Did You Know today?
No.
The answer is no, I didn't.
Malaria are called hemosporidian parasites.
They are.
In all our TWiPs, I never heard you mention
Now,
that.
we've called them apicomplexa
Right.
because that's a real taxonomic group, but hemosporidian are organisms that are related to they're not related to microsporidium at all. They produce sporozoites during their life cycle, right?
Mm-hmm.
Because they're produced inside the mosquito, and they get inside the blood. So that's hemosporidian.
Hemo is the blood
Yep.
reference. I see.
Yep.
And these are the
right.
malaria
That's right.
parasites.
As opposed to trypanosomes, which are called
They're not sporidia, right?
no, they're not.
But they are in the
what
blood.
are they?
Hemo. I don't know.
See, this is called Stump the Chumps. On Car
The
Talk,
chump.
they have that all the time.
Really?
It's called Stump the Chumps, and here we are trying to stump each other, and let's play that game. So they're called hemoflagellates.
Okay.
Hemoflagellates.
So the last part of the word is some aspect of their morphology.
Exactly right.
So for the malaria, it's
Exactly.
sporidia.
Yeah.
For the trypanosomes, was it?
Yeah.
Hemoflagellates.
Hemoflagellates. That's right.
Give me another one.
The Leishmania also, because when they infect you,
Hemoflagellates.
they infect as flagellated organisms, but then they round up and they become amastigotes.
Okay. Now they say here, and you confirm if this is correct. I want you to confirm these things.
Oh dear.
Human infecting Plasmodium species are only a small fraction of the over 550 species in the order Hemosporidia. So it's actually an order.
That's right.
Hemosporidia.
That's right.
With 17 genera.
That's right.
550 species of what you would look at and say this is a malaria.
Yeah.
Right? Wow,
That's right.
malaria.
That's right.
Of course, they don't all call malaria in their hosts, right?
Well, What do you mean?
The typical human disease.
Remember what the word means. It's derived from the Latin, which means bad air, right?
So to a turtle, and by the way, turtles do get infected.
Yes, they do.
Listen, mammals, birds,
right?
Squamate reptiles. What's a squamate reptile?
Squamate. We're going to hear from our listeners. I know we will. And they will, some of them will
Want me to
tell
look
us.
it up?
You could do that now and spoil everything.
Well, you don't know, in other words. I've stumped you.
I don't. I've never heard that term before.
Squamata, or scaled reptiles, are the largest order of reptiles, comprising lizards and snakes.
Scale.
Wow. Over 9,000 species. All right, so that's what a squamate reptile is. And turtles and crocodilians, they're all infected with hemosporidia.
They are.
So you agree with that?
Of course.
They also say one hallmark of all hemosporidian parasites is the obligate host switch between a vertebrate intermediate host and an arthropod vector as a
transmitter of the infection.
Blank host.
As a definitive.
Very good. See, you got it. You
Vince,
get $400.
I used to really?
Your next question.
I didn't get that one, I thought.
I'll take $1,000 for vectors.
Oh, right.
Exactly.
I would love to have.
What is that game called?
It's called Jeopardy.
Can we do parasitic Jeopardy?
We could do that. That would be great.
I think that would be fun. We could actually get contestants and.
I think we should have that at every annual ASP meeting.
Infectious disease Jeopardy. It's
Why
probably
don't
been
they
done.
do that?
It's
and then give out
done.
textbooks or scholarships and. There would be grants. How'd you like to win your grant on Parasite Jeopardy?
All right. So what we've established is that the Hemosporidia is quite a large order.
Yes.
And also bats are the other subject of this paper.
Yes.
They're pretty large
Volate
also. Do you know what the order is of bats, Dickson? You do. That would be in Jeopardy. The word would be, this is the order of bats. What is the order of bats?
Draculae. Draculidity.
Chiroptera.
Right.
Chiroptera.
Chiroptera. It's CHIROAPTERA.
Chiroptera.
What does aptera mean?
No.
It's a wings.
It's the second largest order of mammals.
Right. What is the first largest order?
I know because I see
it's
it
Rodentia.
Here. Yeah.
That's right.
Is that a country in Africa?
It is. It used to be. Now it's called something else.
Rodentia is the biggest, right?
That's right.
So the bats are 1,232 species, 18 families,
right,
and a number that Dickson has thrown out many times over the years. What percentage of all living mammalian species?
About 20%.
Exactly.
Well, I learned that from Peter Daszak, who is a world-famous wildlife pathologist who knows a lot about bats now because he was impressed with how many different species there were as well. And he and I used to co-teach in a course called Emerging Infections,
right,
in which Steve Morris and I were co-directors. And Peter Daszak was an annual visitor.
You should give a lecture in that course.
I used to. I'm retired now.
I suppose.
I get to go fishing more often, remember?
Apparently once a week, right?
Whenever I can.
And you just wake up and decide I'm going fishing.
Well, I more or less determine when I can go based on my obligations.
We've gotten off the topic here.
It doesn't matter. We're intermingling.
All right, so we talked about the bats
stories with. But we don't. Wait a minute.
What if we talk?
Wait.
Go ahead. Okay.
I'm trying to say this is not off the subject because once when I went fishing with a friend of mine, Bob Demarest, who is a world famous medical illustrator, we encountered a bat, a little brown bat in this case, which is a very common bat species in this part of the world, hanging apparently in midair with no particular reason for being there, and it looked as though it had died.
Mm-hmm.
So little brown bats are the ones that everybody likes around their houses because at night, when everybody else is asleep and bats are now awake, they go around the countryside ingesting insects. And in some cases, whenever there are blossoms of mosquitoes, they go and ingest them. So they're insectivorous bats. Some are eating fruit.
What are they called?
Frutivores.
Frutivores. There's a real name for.
I know that. I forgot it, so I just made that up.
I like insectivorous.
Insectivores are
and
great.
What about blood-sucking bats?
those are hemovores.
Hemovores?
Hematophagous. No, they're called hematophagous.
All right.
Like
So so you found this bat, and what's the
bat
story?
was apparently just suspended in midair, but that couldn't be right because the wings weren't flapping. This was in broad daylight.
It wasn't hanging on to anything.
Right.
Got
So
it.
what happened? Now remember, little brown bats can transmit what horrible viral disease, Vincent?
I don't know what the little brown
No, rabies.
Rabies? Okay.
Oh, yeah, absolutely. So
It's
Bob
probably the
Demarest-
only one we know of that is actually transmitted, right?
Correct.
Because all the others,
Correct.
the viruses we have talked about, are
Yeah,
apparently in bats, but we don't really know how they
We
get from
don't.
bats to people.
we don't. But rabies, we do.
Yeah,
know
rabies for
something
sure.
About. That's right.
You go in a cave with rabid bats, you can get rabies. Is that
You
the
can
idea?
get inhalation rabies, which
Or a
is
bat
even
can fly into
horrible.
your home and scratch you.
and bite you. They have very sharp teeth.
Yep, nice pictures of
They have
bats in this
huge
paper.
sharp teeth. Why? Because they have to bite into unripened fruit sometimes, or figs. They're great pollinators, by the way. Bats are
So when a
wonderful
bat is
pollinators.
insectivorous,
You're right. It
what
just
is
eats
it?
insects.
It just picks the insects out of the air?
Some bats eat fish. Some bats
I
eat.
want to know how the bat finds the insects flying through the air with
Oh, yeah,
echolocation.
they echolocate. They echolocate.
So they're flying, they boom like a bird, right? They just
That's
pick
right.
it up.
When I was a postdoc at Rockefeller, there was a world-famous bat biologist there, Dr.
Mm-hmm.
Griffin. And he worked up at the Bronx Botanical Garden and Zoological Society, and helped to develop their exhibit up there called World of Darkness. So if anybody comes to New York City and wants
Still
to,
there?
it's still there, yeah, and it's wonderful. And it shows bats in most of their behavioral aspects. But, okay, so this bat suspended in midair turned out to be hooked by somebody's fishing line, a very invisible thread of fishing line, at the end of which was a fly.
Mm-hmm.
Now, bats eat insects. And at night, the bat can't really distinguish that little fly that someone had taken a back cast and hooked his line or her line on the tree. The line broke, but it broke above the fly. So the fly was actually suspended down over the river. Here comes this little brown bat, catches the fly, and gets caught up in this line. And it was flying all night long trying to get loose, and eventually it ran out of energy.
Did you let it go?
Well, here's what happened. This is an exciting story, actually,
right, I'm waiting.
because Bob and I both realized that the moment we touched the fly line to try to pull it down from the tree, the bat came alive.
Right.
And we realized this is not a dead bat. This is an alive bat.
It's like
A
a
live
virus.
bat.
It's not like a virus.
It's not like a virus, but they carry rabies.
Okay.
So he and I both cautiously thought our way, now what are we going to do? We want to release the bat. So what we did was Bob went back to the car and brought out his pliers, long needle-nose pliers that he has as part of his emergency kit, right? And then I grabbed the line way above where the bat was, and I cut it with my nail clippers, which I carry for fishing purposes. And I carry this bat wriggling around on the end of the line
Right.
to the shore. The bat then rights itself, and you know bats can actually crawl. So this bat started to make its way into the grass. And what Bob had also brought was a piece of paper toweling. So he put the paper towel down over the bat to hold it down. And with the long needle-nose pliers, he reached into the mouth of the bat and he extracted the hook.
Wow.
Now
Is
the
it
bat was
hard?
free to do whatever it wanted. So the bat, and then we released it. And then it just crawled and crawled and crawled off into the brush, hopefully to regain its strength, to resume its normal activity.
Was it hard to pull the hook out?
No, it apparently was not, but it was,
Did it bleed?
we didn't notice whether it bled or not.
'Cause
So
some fish, it's often very difficult because they swallow the hook, right?
this is true. This is very true. So over the next couple of hours, Bob hooked and released a very large trout. It turned out to be a brown trout. So he decided to write it up. This was a wonderful day for Bob. So he wrote an article, which was actually published in the American Journal of Fly Fishing, which was a publication that comes out of Vermont from the American Museum of Fly Fishing. And he called it A Couple of Browns. And one of them, of course, was the brown bat, and the other was the brown trout. It was a wonderful day, but it sort of made us realize that, we've altered nature in many ways. And here that poor little bat was prevent it from carrying out its normal ecological functions.
It's nice that you released it.
Oh, yeah. Oh, we both thought very highly of its activities and wanted it to survive.
Well, I'll tell you a
All right,
fish
now
story.
back to our real story.
No, I'll tell you a brief fish story. We were fishing off the dock at the shore a couple of years ago. My son cast out, and he hooked a seagull that happened to be flying by at the same time in midair. So the gull sat in the water about 100 yards away, and every time my son pulled the line, it would flap.
Oh, gee.
So I said, what are we going to do? I said, I have no idea. Every time we tried to pull it in, of course, the
struggling,
gull.
Of course.
So we just cut the line, and we figured eventually. It was like in his wing or underneath.
Dear.
It was probably going to come out at some point.
Let's hope.
And what else could I do? I couldn't get the seagull. He was a fully functional, large adult. Do you know how I knew it was an adult and not a juvenile?
How did you know, Vincent?
It was white and gray, not brown.
Okay.
Because the juveniles are brown.
That's it. That's
okay,
right.
back to the story. Bats. So we have lots of plasmodia or hemosporida. We have lots of bats.
Yep.
And there's been evidence.
And we have lots of rodents.
Yeah, we do, but they don't figure.
Yeah,
Actually, they do figure. I'm sorry. I am so sorry.
That's okay.
Why do I speak so quickly?
I don't know, because you just like to. But remember, rodents arose before bats.
They did.
So the earlier harbingers of bat parasites had to be
rodents, probably.
Rodents. So
and they're going to provide a little evidence for that.
They sure are. They sure are.
Now,
so parasites can lead you to find out things about your hosts that have nothing to do with the fact that they have a parasite, but rather that they might be related to something that had the same parasite.
And so the other issue here is that apparently it's been known since the late 19th century.
Yes.
Which would be 18-something, right?
That's right.
That bats have hemosporidia, but they haven't been well characterized. So, that's the scenario for this paper, right?
Yeah, exactly right.
Would you like to say anything else before we move on? All right.
Nope.
So they went to, this is to finally answer your question of an hour ago. Where did they sample? The Upper Guinean forest ecosystem.
Wow, that's pretty exotic.
That's in Africa, right?
Pretty exotic.
I've never heard of it. They say it's one of the most biologically diverse and endangered terrestrial ecosystems.
Right.
Do you know anything? I know you're an ecologist.
Yes.
Do you know anything about this Upper Guinean forest?
No, I don't.
Okay.
I don't.
Then we'll have to just stop there. They captured bats. They captured 274 bats in this forest ecosystem. They have a little map of Africa here, and they show Guinea. Is that how you say the country? Guinea?
Guinea, that's right. So it's Ghanaian. So there's a worm that's named after the country.
Ghanaian, the Ghanaian forest, the Upper Ghanaian forest.
That's right. So there's a guinea worm. Do you know the guinea worm?
Yeah, we talked about it.
We did.
Sure.
Guess where that's from?
Guinea?
No, it's from Guinea.
It's from Guinea. And they show on this map Guinea,
right?
Côte d'Ivoire,
right,
and Liberia, which is where a lot of ships are registered.
Do you know why it's called Liberia?
Why?
Because that's where, after the Civil War, a lot of slaves were given the option of going back to Africa, and this country was created for them, and it was called Liberia because they were liberated.
You know, a lot of ships are registered in Liberia.
I do know that.
Probably because it's cheap to do
that. In fact,
that.
Liberia, I think they still use American money as their currency.
Now, these bats were captured a long time ago, between December 2006 and December 2008.
Right.
And 2010. So what they're doing in this, they're actually looking at blood, and you'll tell me about that in a minute.
Sure.
But what did they do, freeze it?
I guess.
And you can still look and find malaria parasites in the blood?
I hope so. Well, they might.
Don't the red blood cells break?
No. They may have made blood smears at the time of capture also.
And those are preserved? They put them on a slide, and they seal the slide with nail polish,
you fix it first in methanol, and then you stain it with a stain which differentiates the nucleus from the cytoplasm.
Like Giemsa?
They're Romanovsky stains.
Romanovsky, all right.
Like Giemsa or Wright's. And then Giemsa is much better, but it takes a longer time. And
H&E?
Takes more time.
Okay.
What?
H&E, hematoxylin and eosin?
No, they don't do that one at all.
No?
Nope.
That's what we use for...
That's on tissue sections.
Tissue, yeah, okay.
Yeah, but they don't do that on blood smears.
Got it. All right, 274 bats.
Right.
and they looked through the blood of each one of
Correct.
thin blood smears, they call it.
Thin blood
So I
smears.
bet you're right. They
Wait.
probably took them way back
There's
when,
another. What if they call them thin? Why would they call them thin blood smears?
as opposed to thick.
That's correct.
But a blood smear is by definition thin,
yeah,
right?
it is, isn't it? So what is this thick?
Well, thick tissue section would
No,
be
no,
thick,
no,
right?
no, no, no.
Now tell me what thick
I
would
mean,
mean.
this is great. This is absolutely
Because this is
great.
up your alley. You used
It's
to
right.
do this
This
stuff.
is what I did for a living when I first
So you
started.
could still be valuable.
Well,
You
I
have
think
to
that's
say,
still a matter of opinion.
I
but-
think you should say, I am still valuable.
Well, I feel valuable to myself, at least, of course.
Defend yourself. Come on.
Okay, so to do a malaria examination on people and quickly, all right, it's almost routine. You take a drop of blood and smear it with another slide
Right.
onto a clean slide. It has to be clean.
Right.
And at the end of that, where you feather out the red cells into a single layer, just after that, you'd place a drop of blood of the same patient and just leave it there. And that's the thick part of the smear.
I see.
So then when you stain it all up together, you know what you're going to get? You take the thick part and dip it in water first after you fix it.
Well, first you have to fix it.
Well, no, you don't want to fix it first, because if you do that, then it won't work. You have to lyse open the red cells,
How do you do that?
and you do that by creating a hypotonic situation.
But if you put it in water, all the cells are going to wash off.
Well, no, they're not after they dry.
So you dry it first.
You dry it first.
So now and how long does that take?
It depends, but if
An hour?
A couple of hours.
So you put the slide on the bench top, you let it dry.
Yeah. And then,
And then you
the
dip
drop
it.
of blood end you stick into water, and you lyse open the red cells,
Mm-hmm.
and you get rid of all of the hemoglobin. But the ghosts and their parasites are still
Yeah,
there.
cool.
And then you dry it down again, and then you fix it in methanol and stain it with the stains, and then the first place you look is on the thick smear. That's a quick examination. And if you see parasites, because they accumulate in large numbers because they're stacked on top of each other, then you can go to the thin smear and actually survey that feathered part of the smear where the cells are just barely touching each other, and you look for the specific species of parasite.
What form are you looking for in the blood?
Well, you're looking for the forms that infect the red cell.
So this is for the hemosporidians, the morphology is clear, right?
Oh, absolutely. And you can differentiate them based on their morphology. You don't have to do a DNA examination, although they do have modern tests which will actually do that.
So they went through 274 specimens.
That's a lot.
How long do you think that would take?
Depends on how heavily infected each one is.
Yeah, they say later they weren't very heavily infected.
No, so they have to look a long time. In the laboratory for human malaria, the rule is if you look for 20 minutes on a blood smear and you don't find any parasites, you end up saying no parasites seen. You don't say negative. You just say no parasites seen.
Because if you had just done one more minute, you might.
That's right, or another hour. So you take smears every 6 hours, and if
see.
you do that for the first 2 days of a patient, you'll usually detect it.
So they found parasites in 111 bat specimens.
Remarkable, though, isn't it? That's half.
40%. Yeah.
Almost half.
And then they confirmed it by PCR.
That's incredible.
And they were all positive by PCR. So there were no false positives,
How about that?
which is pretty cool. I wonder if they had screened them by PCR
Yeah,
and then
you may have had more, even.
Yeah, who knows?
Who knows?
Well, actually, the ones that were negative by microscopy, they tested by PCR, and they were negative. So
Interesting.
very low,
microscopy is pretty good technology.
very low false positive rate here. Not zero.
What's interesting is that if they couldn't do the PCR in 2006. I don't
No.
even think the probes were available then.
For these malaria
Exactly.
genomes. Yeah, they didn't know the sequence.
Exactly.
That's a good point.
Yeah.
So maybe they just decided now we could do this study because they want to get sequences to do the phylogeny,
But
right?
of course.
All right, so the infected bats were in 5 families.
Right.
Pteroptidae. No,
Yeah. I want to watch you pronounce these names now.
Pteropodidae. No, Pteropodidae. Vespertilionidae. Miniopteridae. Hippocidaridae and Rhinolophidae.
Excellent.
I know the Pteropidae. Those are the fruit, the flying bats or the fruit foxes.
Oh, you were familiar with that.
The Vesper bats.
And what virus do they usually carry?
Well, they have lots of viruses, but
What's the one most famous, though?
Hendra and Nipah.
Nipah. The Nipah, yeah.
Now, I recently read a paper on Hep B found in bats, hepatitis B virus found in bats. And it was found in the Hippoceridae and the Rhinolophidae, horseshoe bats, and I forgot what the other one is called colloquially. Anyway, they have parasites, malaria parasites in these five families.
Yep.
And then they sequenced. They did a lot of classification based on morphology and hosts, and they sequenced the genome as well. And then they can construct a phylogenetic tree relating them.
Absolutely.
Now, did you know, I didn't know this either, here are
Go on.
four genera of, I keep forgetting the name, Hemosporidia.
Hemosporidia, yes. Go on.
Plasmodium, right?
Yes.
That we all know.
Of course.
Here's a new one for me, Polychromophilus.
Right.
Did you ever hear that one before?
Nope.
Nycteria
Nope.
and Hepatocystis.
Hepatocystis I've heard of because it also infects monkeys, as you'll notice.
So these are all, aside from the Plasmodia, which contains the human malarial parasites, right?
Right.
These must be...
Polychromophilus looks like it only infects bats.
well, according to the picture, right?
Nycteria, it only infects bats. Plasmodium vinceri infects bats and mice. And by the way...
Hepatocystis does bats and monkeys, or primates, I guess, right?
Yeah, I'm interested in knowing what effect body temperature has on susceptibility, though, because...
To Plasmodium?
Yeah, because
Or
I
to
know that
malaria,
rodents
yeah.
have a higher body temperature than humans, and so do bats. But bats, when they undergo estivation, that is, resting during the daytime, does their body temperature drop? And so I think some bats hibernate as well, and so that their body temperatures certainly do drop.
Right.
And I wonder how that would affect the outcome of these infections, that's all. Just a curiosity. Again, I just think some of our listeners may even know the answers to that.
So I'm going to just mention a couple of other interesting things.
Please.
Before we talk about these phylogenies.
Right.
They had very low parasitemias.
Yes,
Now, you could tell that by looking at the smears, right?
of course.
And they say they're only mature gametocytes.
Ah.
What does that mean?
This is very interesting. It's possible that these parasites in bats mature very fast and then stay around a long time as the stage which is necessary for the vector to acquire in order for them to become infected. So it's fascinating because in most human infections, the development of gametocytes occurs later on during the infection. Right away, they want to build up numbers, and nobody really understands the process of whether the parasite goes on to reproduce inside the red cell and eat up more red cells, or stop reproducing and develop to this pre-sexual stage that it's necessary for the mosquitoes to acquire in order for them to acquire their infection. So these bats all had only the mature stage of the infection and no intermediate forms of, like, say, ring stage or trophozoite stage or the schizonts. None of those stages were seen, only the gametocytes. Now, you can't tell that by doing a PCR because the DNA is going to stay the same all the way through. Right. So, but when you look at the smears, all they could see were the stages that the mosquito needed to acquire to retransmit this. That's fascinating stuff.
Do you think these bats were sick?
No possible way. No way. Bats just don't seem to get sick.
Maybe the ones you capture are sick. Well, that's why you capture them.
These guys, I mean, this paper emphasized the fact that they're immunotolerant to a lot of things.
Right. Yeah, they
which
probably
is fascinating. Wouldn't you like to know why? I mean, if I were an immunologist, I'd be studying bats.
A few labs globally are beginning or have begun to study bat immune systems.
Why don't they die from rabies?
Exactly. It's a very interesting problem. And this is worth studying because...
No kidding.
Maybe we could learn some things that we could apply to ourselves.
No, we could never... No,
of course,
don't be silly.
Just understanding is worth it in itself, but it could have a benefit for us.
If you were to explain this to somebody like a senator or a congressman and say, we'd like some money to study immune systems of bats, I think they would just
Well, that's why
ask their aides to come out and lead you away from their office.
We've recently learned that they cannot be trusted to make decisions, especially fiscal decisions.
And what are we going to do
science?
about
They
that?
can't. No, we can't ask Congress. Although the NIH has to ask Congress every year for money, and they probably say, "Well, what are we getting for your money?"
Exactly. What
That's
they
the
say.
problem with science.
What are you getting for? Look at what you could get from this. This is a treasure trove of
Treasure.
new approaches.
It is fabulous.
It is great stuff.
By the way, Plasmodium
Yes.
were only found in fruit bats.
How about that? How about that?
That's interesting.
It is interesting.
And insectivorous bats. Okay, fruit bats and insectivorous bats.
Go on.
What else do they say? Nycteria were found in horseshoe bats, which is rhinolophus, which
Yeah.
also have hepatitis B virus.
Right.
And Polychromophilus is restricted to insectivorous bats. All right, that's just of interest
And
because
this may relate to their behaviors more than their susceptibilities, because it's where they fly to get their food as to the availability of vectors. And it's also where they roost at night. Fruit bats roost right out in the open.
yeah.
How do I know that? You've been to Australia. So have I. Did you
I've
see
never
fruit
been
bats?
to Australia. I'm going
I
next
thought
year.
I'm sorry. I take all that back. But you're going to Australia.
Going next July.
You
yes.
will be absolutely stunned if it's still there. By the way, it's on fire right now, so
Australia's on
it
fire.
may be gone by the time you get.
All right, well, I can save plane fare.
Yeah. No, but I hope you get to go because you'll get to see fruit bats.
The vast majority of parasites were hepatocystis. So most of them were hepatocystis.
Hepatocystis, which is a
Liver, huh?
liver-specific hemosporozoan.
Right.
But yet, all the other plasmodia have liver stages.
All were found in fruit bats.
Interesting.
So they found mostly hepatocystis, and all of the hepatocystis were in fruit bats.
Right.
What else here?
So patterns emerge. I wish they had also done a little bit more description on the vectors that were responsible for transmission.
We'll talk about that in a minute, but they don't know too much. Hepatocystis appears to be derived from the mammalian Plasmodium parasites.
Interesting.
So they construct a phylogenetic tree
Yep.
where the relationships among the different what is it again?
Hemosporozoans.
Hemosporozoans can be displayed on a tree, and you can see that hepatocystis is
Yep.
branched off from the Plasmodium. They both have a common
So
ancestor.
leukocytozoans are the oldest group, it looks like.
Yes, very old.
All the way down to the hepatocystis group, which is the most modern.
Most
of
modern.
those.
And
According
before
to
that-
I guess, what are they using as a scale of measure for time?
So that's the horizontal scale on the on these
relatedness.
phylogenetic trees.
Right.
Right. It's a calculated. Well, the horizontal scale is time, right?
Yep.
And it's
Yes.
based on a molecular clock of mutation. And here,
Usually
the
by-
scale is 0.04.
Right. I often wonder about that because I'm sure other people are confused a little bit when they look at these kinds of diagrams. They wonder, what are they basing the relatedness on? And of course, it's a complicated formula of using mutation rates and then trying to find out which ones mutated from others, right? So they go backwards in time usually to the original one, which has the fewest changes, or it has the most changes.
So the horizontal scale is evolutionary lineages changing over time. And
Over
the
time.
number, in this case, which is 0.04, it's the amount of genetic change, that is nucleotide substitutions per site, that is the number of changes divided by the length of the sequence.
Okay.
Okay, so that is in this phylogenetic tree, we have 0.04, which is the amount of change.
But that's their genome, right? That's not their mitochondrion.
Is the genome of these guys, yeah.
How would they distinguish those two from each other?
Well, you could separately, you'd have to separately purify mitochondria, right?
You would. They
Might
have
get
very
mitochondrial
odd
DNA.
mitochondria.
Didn't Plasmodia do, or
Yes, really odd.
hemosporozoites, hemosporozoans?
Right. If you look that one up on your Funk and Wagnall, as they used to say.
I'm trying to see what they sequenced here.
Right. It had to be whole genome sequence.
Oh, here, 4 signature genes representing the 3 parasite genomes were selected for phylogenetic analyses. So mitochondrial cytochrome B and cytochrome oxidase L. Plastid, a couple of genes on plastids as well.
Right.
So both mitochondrial and plastid sequences were used to construct this tree. To answer your question. Why would you do that, Dickson?
Why would I do what? Ask a question?
Why would you do both mitochondrial and non-mitochondrial?
Well, you'd like to see whether...
And what's a plastid?
Yeah, what is a plastid?
You don't know what a plastid is?
I use those terms when I talk about plants.
A plastid is a major organelle found in plants and algae.
Plants, that's right.
Don't.
Plastids and plasmodia have something in common.
What's that?
Plastid sequences. So one of the papers that I once saw delivered at a meeting was the first revelation that Plasmodium falciparum had some unaltered plastid genes. Unaltered. So when he showed his paper, he had actually allowed his mice to swim in water that had a green dye in it. So when he showed the mice, they were all green, indicating that their Plasmodium infections were plant-like. And it was a funny way of presenting it. Everybody laughed, and then they got the idea. You never forgot something like that because of its graphic presentation.
So the apicoplast is basically a plastid.
Plastid.
So that means it has
plant-like genes.
It's got genes in it.
Yeah, but
and then they have a regular.
Those are
genome.
extranuclear genes.
Yes, exactly.
Just like the kinetoplast.
Does that make sense?
Sure. The kinetoplast.
And then they also amplified chromosomal genes.
Right.
From these, then they compared all of them.
Right. So cytochrome oxidase is usually pretty standard stuff. What other genes do they use of interest that are pretty standard in most organisms?
Actin.
Yeah, exactly.
Dihydrofolate reductase, a histone.
Tubulin.
Ubiquitin.
Yeah.
A inosine monophosphate dehydrogenase. Kinete surface protein, P25.
Kinete surface protein. Now, you wouldn't find that in many mammals.
No.
In fact, none.
All right. So a couple of things here. Here's the interesting, some of the interesting things in addition. How do these guys get infected? How do they transmit among them? So they say all 4 Plasmodium species naturally infect thicket rats, African thicket rats, which are arboreal. Why don't arboreal. Why don't they just say they live in trees? Okay, arboreal, and they say that maybe that's how the bats encounter them.
Maybe.
Okay, so all 4 of the murine Plasmodium species infect these thicket rats, and maybe that's how they got into bats.
Perhaps.
But the bat's not going to eat the rat, right? It may feed.
There are no, as far as I know, there are no mammalianivores.
But the vectors, the dipteran vectors, may go for the thicket rats, and the bats. And the bats may then eat them,
they're
right?
Promiscuous feeders.
But they say here the roosting behavior of the thicket rat may attract the same dipteran vectors as those seeking tree-dwelling rodents, which is maybe how the parasites got from rats to rodents. And then bats would eat the vectors, right?
Yeah, well, that's right.
The insectivorous ones.
That wouldn't give malaria, though, would it?
Oh, if you ate it, it wouldn't give you the parasite?
No.
So you'd have to be bitten by the vector?
You would. So that means that the mosquitoes that seek out their hosts by odor or body temperature have little or no distinguishing abilities when it comes to telling a rat from a bat. In fact, their sign says, if it's spelled with a P or an R, we don't care as long as it's got blood in it. So bat, it's trading back and forth. So it's very interesting because some mosquitoes will bite only one group. Like, for instance, there are mosquitoes that will only eat the blood of certain non-human primates, for instance, and others which eat the blood of lizards. They will never attack a human being. And we can see this, by the way, with a virus infection called West Nile, where you've got enormous range of feeding behavior with regards to this mosquito species that also gives it to humans. So, and then some others that only feed on certain other groups of, let's say, rodents or reptiles or that sort of thing.
So they feel that the Plasmodium species in particular are, of bats and rodents are very closely related, and they think that they switched back and forth over the years between the two.
Could happen. It happened. In fact, it did happen.
They also say that they think bats were the first mammalian hosts of Plasmodium before rodents and primates.
Really?
Yeah.
So then they have to say where it came from.
I don't know where it came from. We don't know, yeah.
Well, we know it's a definitive host of the mosquito,
maybe
or parasite of the mosquito, rather.
Yeah. But maybe some other reptiles or crocodilians or some other species it then crossed over into.
They would have to have a very warm body temperature to match the conditions that these animals have. So maybe it was a mammal, other than a rodent.
Another thing here which I don't understand.
Go on.
Is so they say that tolerating high prevalence of parasites, along with high parasitemias, as shown for the hepatocystis infections, might be another example of the well-adapted immune responses of bats.
Well-adapted.
It is possible that even the change in the parasite's life cycle away from erythrocytic schizogony in 3 of the bat-infecting genera could have been an evolutionary compromise of using these metabolically active hosts and driven by the immune responses of the bat. So what's erythrocytic schizogony?
Okay, so there are really 2 stages to all of the malarial parasites that we're used to discussing, all of the human forms, or the primate forms, I should say. When a mosquito bites and injects the sporozoite stages into the blood, they have receptor molecules that allow them to attach to their ligands, they have ligand molecules which allow them to attach the receptors in the liver only. So they can circulate forever until they eventually end up in the liver. When they do, they attach. And that's when they start to initiate the liver stage of the infection, okay? So there's this, it's called cryptic phase of the infection, or pre-patent phase of the infection. The organism disappears off the radar screen. And it goes into an hepatocyte. Do you remember the one that stays around for a long time? They actually don't even reproduce, most of them. They develop something called a hypnozoite, which is characteristic of Plasmodium vivax and Plasmodium ovale. Both of those have these stages that can relapse over time, over a 5-year period, perhaps. So some parasites, what they're suggesting is that some of these parasites have just given up reproducing in blood altogether and just stayed in the liver and just stayed there and reproduced.
That's what they're suggesting here.
The suggestion is
because of the immune response of the bat and the high metabolic activity may have forced them to do that.
Yeah, well, that's highly speculative, but is the liver any less a responsive organ in terms of immune system functions than erythrocytes? Erythrocytes have no nucleus. They have no innate defense mechanisms. They have no...
Yeah, I think the liver can be certainly a wonderful target of immune responses if it's infected. That's why the liver degenerates in part with many virus infections, right?
Well, here.
So,
foie gras.
Foie gras. So they say it's really important to sample in biodiversity hotspots like this.
How about that?
I agree.
Okay.
Did you know that bats originated 66 million years ago?
That sounds like just after the meteor hit.
Probably in Africa, they say.
Or just before the meteor hit, sorry.
And so maybe shortly afterwards, they picked up some malaria-like parasites from an animal that was living at the time, right?
I mean, when I think about ancient times, like what you just mentioned, the benchmark for me is 65 million years ago,
right?
Which is the KT zone.
That's where the meteor hit the Yucatan area, right?
So that's where it did it. That's
water
right.
there.
Yeah.
What's that bay called? The
Gulf of Mexico.
The Gulf of Mexico. Did it hit? Actually, hit off the Yucatan.
It hit just off of where the Yucatan is now.
And you can go under the water and see the circle impact, right?
It's amazing. It's just amazing. So that presumably wiped out most of life on Earth. So if bats arose a million years before that,
Except little mammals.
they were tiny little mammals.
And-
Well, they had to arise from rodents, right? That's what we said.
And some birds were left over from the dinosaurs, right?
Birds, the little ones. Everything little survived
Huh.
because they could hide out.
66 million years ago. Wouldn't you love to have a camera from then?
Absolutely.
Where I could go back.
Absolutely. Well, that's what Jurassic Park's all about.
Anything
bit
else we should do before we move on, Dickson, Despommier?
None, because I think paying attention to articles like this has great merit for introducing to those that are unfamiliar with parasitism or parasites the value of studying them.
Yeah, sure.
Because they show you interrelationships of things that you had no knowledge of before, nor could you have any knowledge of this. But rather by looking at which parasites they acquire and where they live, it gives you a much broader view of life on Earth. I've never seen a good program like the ones that Sigourney Weaver was the announcer for Life on Earth, which is that series, or the ones that David Attenborough was so famous for. I'd never seen one that takes up life from the perspective of parasitism and then goes back and looks at all the relationships that are possible. But wouldn't that be a great show to see?
Should make one, Dickson.
No, I'm not capable.
You're not?
Nah.
Why not? You don't have the energy.
Where would I start? I mean, how you'd need a team of anthropologists and paleontologists and all kinds of other people to get together, and biologists, of course, to look at this data. But this data is just recent in its emergence. That's the problem.
Yeah, you get a few people to talk about it like we do. And
Right.
you just take movies in Gabon.
We
not
need
Gabon.
more
What's the
of
name of
this.
the
We need
country?
more of this.
Guinea.
Guinea.
So the next topic was stimulated by an email from Blaine, who wrote, "Hi, TWiPies.
TWiPies.
I saw today in the New York Times that a hookworm vaccine will be tested in Gabon." That's why I said Gabon just now.
And what do we know about Gabon already? We know that there's a viper named after it.
Where is it?
It's in Gabon.
No. Where is Gabon?
It's in West Africa.
Western Africa, which is near Guinea.
Yeah, it's close.
I found this very intriguing, as your discussions of parasitic worms have rarely included the possibility of vaccines. Can you please comment on this? Are there certain biological challenges to making vaccines against worms, or is there simply no money in making these vaccines since they are not needed in rich, developed countries? It seems to me that it would be much cheaper and simpler to just educate people on how to avoid most of these parasites than to produce a vaccine. Simple practices such as digging your outhouse 6 feet deep or curing your feces before using it as fertilizer would eliminate many of these parasites. I would love to hear your comments on this matter. Blaine is from Alberta,
Cool.
Canada.
Okay, Blaine.
So let's first mention that the New York Times article is called "Hookworm Vaccine Will Be Tried in Africa" by Don McNeil, and it is about a vaccine effort spearheaded by Peter Hotez, your buddy.
My buddy and a former TWiP.
Yeah, he was on TWiP. I didn't realize he's director of the Sabin Vaccine Institute.
Oh, yeah.
I've corresponded with them.
Sure.
They listen to TWiP.
Sure. I bet you listen to TWiP also.
And this trial will start on a few adults in Gabon.
Right.
And it may take 5 years. And Hotez said he began working on the vaccine as a graduate student at Rockefeller 30 years ago.
This is correct.
Did you know him at Rock?
No.
He came after you.
Yes.
He's younger than you.
Yes, he is.
So the vaccine is against
Everybody's
two enzymes.
younger than I am.
No, that's not true. The vaccine is against 2 parasite enzymes that are found in their gut. One that detoxifies the iron and another that digests blood proteins.
Right.
Have we talked about those processes? Somehow iron rings a bell with parasites. That's what malaria needs to get rid of, the iron in the heme and the hemoglobin, right?
Well, it's not the iron that's toxic. It's the porphyrin ring.
What does it do with it?
It stacks it up.
It makes what out
It makes
of
a crystal out of it.
Got it. All right, so we never talked about this. Why would making a vaccine against the gut enzymes of a worm be inhibitory?
Because then you can prevent them from feeding.
But they would ingest the antibodies?
Yeah.
Are you serious?
Sure. It comes in through whole blood.
The worms are in your intestine. Give me a brief review
of hookworm.
Yeah,
Sure.
yeah,
The
yeah.
adult parasite.
How do you get it?
You get it by stepping on it or just sitting on it.
Okay, right.
It goes right through the
That's why
umbilical
you
skin.
dig the
That's right.
outhouses deep.
This
right.
is correct.
So they don't come out of the feces up. Okay.
No,
So you step on it, it burrows through your feet in a
In
cut
fact,
or
if you have
something.
a picture of my new book called People Parasites.
I do.
Let's
I'll
see what's
get
on
it.
the cover.
A hookworm.
Exactly right. It shows their teeth.
Where's the copy of your book?
I don't know, Vincent. I thought I gave you 100 copies just to
No,
keep
you didn't give me
around.
100.
That was a joke.
Apparently, you haven't even given me one.
That's hard to believe that. That's not true. I disagree with you. I know I gave you one. But at any rate, on the cover, we chose that parasite to show how vicious up close these things really look. So the adult parasite actually gloms onto a villus, okay, and chomps down on it.
In your gut. Yeah,
in your
villus.
gut,
Yeah.
And it eats the villus tissue. And at the same time, because each villus has a circulation associated with it, a lymphatic vessel and a blood vessel, it has access to your open blood supply.
Oh, okay.
And it starts sucking your blood.
We must have done this on a TWiP.
We did. Of course we did. It was an
Nicator
early-
Americanus.
Exactly.
What's the other one called?
Anselostoma duodenale.
Oh, it's the Italian one.
It's the
Yeah, I
Italian.
remember.
That's right.
I got to look it up while you talk.
Okay.
I can do this because I am Italian. I
We
can
are
make
not-
fun.
Hopefully making fun of the Italians.
But I am Italian. I can do it.
But we are, doing a very poor imitation of an Italian accent.
But a duodenale.
Duodenale.
Ah, I want to find the name of that episode. I wonder how long ago it was. It was actually...
It was like number 3 or something. No, wasn't that early.
Was
it was
was
like
it-
number 14 or 12 or something like that. Did we do the worms? We did the worms first.
No, we did worms first.
So it might have been like 6 or 7. We did it along with strongyloides.
Really?
Yeah, we did. We did.
Tapeworms, malaria. Then we did malaria after the tapeworms for some reason. Then we did toxo, leishmania, TWiP, giardia, and
Yeah,
amoeba.
go back.
No.
Go to Ascaris.
I think we did it.
No, go to Ascaris.
Really?
Yeah, go to Ascaris.
All right. Ascaris was... No, it's...
4 or 5.
Racaniello is 4. 5 is nurse cell. 6 is tapeworms. It's not a tapeworm, right?
No, of course not.
No. We didn't do
Look
it
at
until
us.
later.
Look at this, up to episode 62 and we can't remember anymore. This is bad.
Yeah, I know, but we're going to get it here. We did Trichuris,
There.
Ascaris.
There you go. And then after that, hookworm.
Oh, so it's not Ascaris, right? Ascaris, then we did hookworm. There you go. It was
Bingo.
called hookworm. And we have the nasty looking picture. Nicator Americanus and Ankylostoma
Duodenale.
duodenale.
That's what
I
I-
love that name.
I wonder why.
So it's drinking your blood. So you're saying the
It is.
antibodies could get into its
course.
gut,
Yeah.
prevent it from
Digesting protein.
and detoxifying iron.
Bingo.
What is this detoxifying iron all about?
You'd have to ask Peter that. I think he's an expert on that area. I don't know, but I think that's part of the biology of this parasite.
Okay, I'm going to put a link to a nice article by Peter
Good.
in Vaccine of this year called The Human Hookworm Vaccine.
Superb. Well, now you remember, of course, he was using some other proteins, derived, cloned proteins,
So two
From-
recombinant antigens,
from the larva.
and one is
It's
expressed in insect cells and the other is expressed in tobacco plants.
Really?
Yes.
Okay.
And they are purified and mixed with an adjuvant.
Okay.
And they make anti-enzyme antibodies that will reduce blood loss and the number of hookworms attached to the gut.
Right.
So it's going to be tested, and then they want to transfer the technology to the Oswaldo Cruz Foundation so
Nice.
that they can manufacture it. It's a
Very
big
nice.
problem in Brazil.
It is. It's a big problem in a lot of countries that are in the tropics.
One of the most common neglected tropical diseases, a leading cause of iron deficiency anemia in low and middle income countries.
And
Did we talk about that on
we did,
TWiP?
of course. It's an
Okay.
endocrinopathy in children too, because it prevents them from thriving. So it's, even though we don't understand the nature of the endocrinopathy, we do know that it does create that. So there's a good reason for wanting to do it. But I understood, though, from an earlier vaccine attempt in China, that the vaccine itself induced a hyperallergic response, and therefore they ceased trials.
Really? This Peter Hotez vaccine?
Yeah, that's right. So there may be different antigens involved in this one.
All right, so Blaine, she says, why not just prevent infection? Why do you have to make a vaccine?
Well, prevention is a great idea, and it works in some situations where you have stable governments, for instance, and seasons in the year, and sanitation. That's the big issue, right? So without sanitation and without seasons, you have universal transmission. It just goes on all year long.
Let me give you an example. Polio is spread by poor sanitation in part.
Right.
In many parts of India, there's poor sanitation. Would you agree?
I would.
Yet we've eradicated polio from India by vaccination.
We have.
There's not been any polio for almost 2 years.
Tell me.
And that's because a vaccine is effective when you can't re-sanitize the whole country.
Is that the live virus or the dead-killed virus vaccine? I know that it's
Sabin
live.
vaccine. Sabin vaccine, the oral infectious vaccine.
So it has a chance in nature to actually come out again.
Well, it's in fact there in India. The infectious vaccine is in the sewage.
And they look at the sewage.
Because
it hasn't caused disease.
When you get it, you excrete infectious viruses.
India's an unusual place in the sense that it's a technologically advanced country with lots of middle-class people. And it's also a country of extreme poverty with lots of poor people, too.
It's the same here in the U.S., isn't it?
Yeah, but not...
Don't we have a lot of poor people?
Not like they do in India. India has a quarter of the world's population.
Yes, I agree that it's more extensive. And
I've been to
probably
India and I've
our
seen the poverty.
sanitation is generally better here.
The poorest person in this country is equivalent to the middle class of India in many respects.
I don't know. I would have
No, I would because I've been to both places.
There. Okay.
And
where in the U.S. are the poorest people?
Appalachia.
Okay.
And inner cities.
Mm-hmm.
Okay.
But they have sewers, right?
They do, and they have television, and we have a lot of things that most other people don't have. And, another thing we have is, get this one, universal health care insurance, which we just got, and a lot of people are trying to say we don't.
So she asks an interesting question. Is there no money for these vaccines since they're not needed in rich countries? So you wouldn't need this vaccine in the U.S., right?
No, but there's still money for polio, even though we don't have any.
But that's because of philanthropy, right?
Yes.
And I'm sure Peter Hotez has to raise money to... Does he use Gates money to do this?
Yeah, a lot of it comes from Gates.
It would not come from pharmaceutical companies, right?
No, because
they're not willing to do this.
They're profit makers. And
gee, who said that recently?
My friend Chuck Innersh, actually. I mean, that's what they were founded on. The pharmaceutical industry doesn't owe the world anything. Period. They are in existence to make money.
Yeah.
That's how they started, that's how they developed, and that's where they find themselves. If the government wants to get involved in making vaccines and drugs, they will have to recreate the pharmaceutical industry. And apparently some governments have taken it upon themselves to support such places, and Oswaldo Cruz is one of those places. So we need some place like that too. We don't have a place like that, although we claim we do. I think NIH-supported research generates lots of information as to how to go about making vaccines and drugs. But once the information is there, someone else has to pick it up and run with it. And so even though large pharma, big pharma as it's called, refuses to do that for certain areas, like in this case infectious diseases, there are boutique industries out there that will pick this up and develop it. And even though they've only got one or two products in their pipeline, as they call it, if those products make a lot of money for those places, compared to a place like, let's say, Pfizer or Merck, which has to have $10 billion of expected income over the next 5 years in order to justify the research, boutique industry, they don't deal in those kinds of numbers. They're in hundreds of millions of dollars. That's enough to support the 30 employees that they have. So there is some hope that smaller industries will spring up with committed people that get support grants from, let's say, the Gates Foundation or the Vaccine Initiative Group that can develop what's really needed most, and that is short-term hits on drugs, that's one thing, and longer-term research on developing vaccines against diseases that are found mostly in places that have poor sanitation.
Peter
So-
says he's been working on this for 30
He
years.
has. He's been working. Essentially, he's been working for 30 years. He hasn't had a vaccine in the pipeline for 30
Right.
years, but he's been thinking about what would I do if I had one, and now he's in a position to actually do it.
So it just went through phase 1 in
Yeah.
the U.S. and Brazil,
Yeah, that's great.
safety, and now it's going into phase 2 in Brazil and sub-Saharan Africa in hookworm endemic regions.
Exciting stuff.
They want an 80%, efficacy in preventing Necator infection.
Well, that may
Necator.
block transmission then, because there won't be enough parasites around in the neighborhood to give it to everybody else then.
So are you saying it could be eradicated?
Not eradicated, but highly controlled.
Who else gets Necator americanus
I mean, who else?
besides people? Nobody else.
Well, I'm not sure. Maybe there are some non-human primates like bonobos or chimpanzees, but I think in general, only people.
Why don't we just use albendazole and mebendazole?
Because there's a growing feeling that we're going to induce resistance if you use it at the community level, and that's the whole reason why we don't do that. And ivermectin too. They've got cocktails for these parasites already that include those drugs. And the problem with them is that once you give those drugs to the communities, let's say there is no resistance. You can't give those drugs forever to these communities. So they come in once a month or once a year or every 3 months, and they give them to about 80% of the children in school. That's how they administer these drugs. And then they come back next year, and they've got the same high parasitemia loads again in the same individuals that receive the drugs. So that means that these parasites on their own don't induce a lot of permanent immunity. And that's the vaccine challenge, to find antigens that parasites are unable to present an immune attack to. So a cathepsin, which might be the protein digesting enzymes of the gut tract of this worm, those may be identified like that. In fact, they've actually identified the same cathepsin types of enzymes that Plasmodia uses to digest hemoglobin into the heme part and the globin they use for their own nutrition. There are some people that would like to inhibit cathepsin D in their parasitophorus vacuoles as well. So
There's a very good article which you should read.
Yep.
They talk about the whole development of this, which was done by the Sabin Institute and the Oswaldo Cruz.
yeah,
They can make it for less than $1 a dose, which is good.
Less than $1 a dose.
But this trial, this phase 2, is really going to make or break. If I don't get 80%,
Tell me about it.
it's it. So, in a way, they're taking a crapshoot, right? Who knows if these are the right
no pun
targets?
intended in this case.
Wow.
They're all crapshoots. Look at the amount of money we've poured into an AIDS vaccine without success. Look at all that money. But we're maybe honing in on an AIDS vaccine that does work. That's why it's called trial and error.
That's why it's called research.
Re-search. I've always called it that. You go back and do it again. If you don't find it, you go back and do it again. You go back and do it again. You just keep doing it until you get it right.
All right. John writes.
Okay, we're getting into some mail now.
Yeah, he sends us a link to a local newspaper article, Zoonotic Research Taking Framingham Man to Peru. Dickson, you know where Framingham is?
It's in Massachusetts.
Very good.
That's where they did the big study.
Let's see if I can
Human
find
health.
this article. So that link there from
Yep.
the letter from John.
Right.
Click on it, and it says it's from Metro West Daily News.
I see it.
And it says this young man, veterinarian, is he's 54 years old. He has a nonprofit company. It's going to stop the spread of parasitic diseases transmitted from animals to humans. He's trying to eradicate pork tapeworms in Peru.
That, through public health education, is possible. I would say it's possible.
It
would
doesn't-
agree.
I'm trying to get to the part where he tells us exactly what he's going to do. His nonprofit is called PAZ.
He's going to educate people on
He's
how
going
they
to
can track
educate
them.
people.
No, that's right. It's done by education. I mean, because if you didn't eat raw or undercooked pork, you could avoid the infection.
So you just teach people not to do that.
Yeah, well, that's good luck on that, because changing
Okay.
eating habits of people are probably that's probably the most difficult thing you can approach in terms of anthropology.
So he has a website. We'll link to that. It's called the Pan American Zoonotic Research and Prevention, and their mission is to prevent the spread of animal-to-human disease through research, education, action, and community engagement.
Right.
Dickson, is this not worth doing?
But of course it's worth doing it. But it requires constant vigilance, and it requires buy-in from the public. So you're going to have to tell them exactly what you're going to do. You have to be truthful with them. And then the last thing that's really important is that you have to monitor it. And you have to appoint people in the community itself as the leaders for this initiative. They have to take ownership of that idea. And if they don't, then you can succeed for a brief period of time while you're in the community, but the moment you leave, they'll revert right back to where they were before.
Yeah.
So you really have to educate to the level of convincing them that neurocysticercosis is a bad thing. And here's why we're doing it now. We're doing it to keep your children from developing seizures when they get to be adults, just like you had, remember? So here's how to do it. And so it's an easy thing. If you drug out all the adult worms with praziquantel, and at the same time change the eating habits so they don't eat raw or undercooked meat, you can achieve your goal. But it's never been done before, so good luck. I'm saying that in all seriousness. Good luck, because maybe you've got a new approach to educating the local communities as to why they should do this.
He's also targeting Lyme disease carried by dogs.
I would be careful of biting off too much.
Yeah. Well, thanks for sending that, John.
Yeah, and he identifies Chagas disease as another one that he want to work on.
Robin writes, parasites may be sneakier than we surmise. He gives a journal article, Transformation of Malaria Parasites by the Spontaneous Uptake and Expression of DNA from Human Erythrocytes.
Wow.
So apparently, in this paper, Nucleic Acids Research, which is a good journal, they have found that the erythrocyte stage Plasmodium falciparum malaria can take up DNA from the host into their nucleus.
You know, Vince, on a larger subject...
Is that good?
Well, no, of course not.
Parasites were transformed to a drug-resistant state when placed into cell culture with erythrocytes containing a plasmid encoding the human dihydrofolate reductase sequence. So in other words, DNA can go from erythrocytes to Plasmodium. Horizontal gene transfer, right? Not surprising.
Not surprising.
Just like it can go from your gut bacteria to you.
Or vice versa. We'll talk about that. A totally different.
And then he also sent, we may have more baggage than we know. Invasive genes, humans incorporate DNA from a parasite. This is another article.
Yeah.
It's from Science News Online.
Right.
They apparently Trypanosoma cruzi DNA has been taken up into human DNA.
Right.
So people with Chagas disease, the researchers found pieces of DNA from the parasite within the genomes of each patient, specifically at chromosome 11. Wow. Amazing. It's in the cell.
Site-specific.
Hey,
wow.
Why don't we do that as a paper?
Sure.
It was published in Cell in when? When? Oh, that's 2004. That's really old.
I would like to see that confirmed then.
Yes, if it hasn't been confirmed, then it's really hard, isn't it, Dickson?
I would say it's impossible.
And this is an old citation also.
Right.
Anyway, thanks for sending that, Robin.
Right. I'll read the next one. How's that? Jim writes, hi, a PDF made from a local newspaper article is attached concerning a biologist who found a nematode embedded in his mouth. This article is available online from the Daily Press, but you have to subscribe to it to read it. I assume it's okay to make the PDF available to your listeners if you desire. Regards, Jim. Well,
all right.
You know, I did see this, actually. I forget where I saw it, but maybe, Vince, you sent it to me?
Yeah,
and said, my goodness, how can you possibly have a worm coming out of your mouth? And
yeah, so
I said, it's Gondulonema pulchrum.
This is it's an article from a local newspaper. It's actually William and Mary biologist removes a rarely reported nematode from his own lip. a rough
Gongylonema pulchrum.
on the roof of his mouth.
Yes.
And so this article is about him saying Gongylonema pulchrum,
Gongylonema.
also known as the gullet worm. Where did he get this from, Dickson?
That's right, Vincent. That's an excellent question, and I'm so glad you asked. Because it actually harkens back to a large outbreak of Gongylonema pulchrum from people living around a lake near Guadalajara, as I recall. And in that part of Mexico, people wanted to feel as though they were not being left behind by the times, so the local restaurants started to offer their own version of sushi. They didn't have access to saltwater fish, though, so they used the fish that they could catch in the lake. Now, you know, sushi doesn't mean uncooked food, because you can order smoked eel and shrimp at a sushi bar, and those are cooked items, by the way, and the rice is certainly cooked, right? But they also wanted to have their own version of raw fish, like tuna fish, which you get at a sushi bar, or yellowtail, or even mackerel you can get at a sushi bar, right? Now remember, when we discussed this when we discussed ascaris, we discussed all of the other ascarid-like parasites, and we discussed the role of sushi in transmitting parasites that live in the gut tracts of sea mammals, and how they get it, and then why we get it, because we eat their fish before they get to them, and the fish weren't clean, and the parasites migrate up into the muscle tissue, and then the muscle tissue is used in sushi bars, right? So here's a parasite that lives in muscle tissue of freshwater fish, that when you eat them, you can develop this parasitic infection called Gongylonema pulchrum. And it could locate in all kinds of weird places throughout your body, including your lip, under your fingers. It can go in a lot of different places. It's not site-specific. It's really a horrible parasite in that sense. And here's, maybe this guy went down to Mexico and had some local sushi somewhere, ate a piece of freshwater raw fish, and developed this weird infection as the result.
He said it's the 13th confirmed case in the U.S. ever.
Yeah, that's right. Lucky him.
He isn't sure how he got infected. But if it wasn't food related, it might have been from drinking well water while he was in Maine.
That's all. In Maine? No, I don't think so. Doesn't make sense.
So he was doing some research in Maine. Water samples taken at the research site showed the clear presence of insect parts in the water. So who cares about insect parts?
Well, the Gongylonema pulchrum life cycle is pretty complicated. If you conjure it up on your video screen, as I would say, you can see that it's got some features. And the reason why we haven't discussed it yet is because, indeed, we've only had 13 cases in the United States, but many, many more cases down in Mexico, by the way, and other places as
60 globally, apparently.
Yeah, wow.
So he wrote a paper. So this guy's a biologist at William & Mary. He wrote a paper. It's been published in this month in the American Journal of Tropical Medicine and Hygiene. Gongylonema pulchrum infection in a resident of Williamsburg, Virginia, verified by genetic analysis. He actually took the worm and he extracted DNA and he sequenced it.
Well, it wouldn't have been necessary, but that's fine.
And he wanted to call the paper, "What's in My Mouth? I Think It's a Worm."
No, I would have had another title for that paper, I'm sorry to say.
What's that?
Is that Gongylonema pulchrum in your mouth? He said, tongue in cheek.
He found this in his lip, and he pulled his upper palate, and he pulled it out.
Yeah.
With a forceps.
It must have been pretty upsetting for him to see that.
He said he got a live worm. It came wriggling out at the end of his forceps, 2 centimeters long.
Now, I have a question for him. What makes him think that's the only worm he caught?
Ooh, Dickson. You're right. So it could have gone elsewhere in his body?
Yeah, sure.
So this guy could be toast.
No, you don't get toast.
Does this kill you?
I don't know of anybody who's died from it, but I don't know very much about the parasite, to be honest with you. Although we could research it and come up with a show on that next time if you'd like.
He
The
said
rarest of rares.
he took it out of his mouth at 3 a.m.
He was sure
sometime during the
happened.
night, and then in his pajamas he carried it to his lab at William and Mary to examine
How
it
funny,
under a
how
microscope. Would you have
Well, that's
done
a
that,
good
Dickson?
biologist. Well, it depends on the size of that worm.
That's a great story. Great story.
And yeah, that's right. So I would encourage everyone who travels to avoid sushi.
Okay. Dixon, take the next one, too, from
Unless
Mike.
you're really aware of it. All right, take the next one, too.
Mike, you still got it there on
I
your
do.
thingy?
Mike. Hi, guys. I know that insect larvae are much bigger than your usual 40.
Forte, our strength.
Then, I see. I'll try this whole thing again. I know that insect larvae are much bigger than your usual forte, but I find the butt fly life cycle fascinating. I don't think it can be defined as an ectoparasite as it actually develops inside the skin, not on top of it. Care to discuss it a little? I would love to hear your take, Dick. Well, yeah.
Do you know what a botfly is?
Shh, of course I do.
I'm sorry to insult you.
That's not insulting. That's just
Want to know.
you're being forgetful over the time. The fact that we discussed this together at some time.
Oh, we did? We did discuss it?
Sure, we did, because we talked about how the insect captures a mosquito and has the eggs of the mosquito underneath its. It lays its eggs under the belly of the mosquito, rather, so that the botfly doesn't have to actually land on the host, us, in order to get its package delivered.
So the mosquito lays botfly eggs on us.
Right, that's right. It doesn't even lay them. They're sort of connected underneath on the abdomen of the mosquito, and when it goes down to take a blood meal, the warmth of your body stimulates the larvae to hatch out of the eggs on the bottom of the mosquito. They fall on your skin and immediately crawl in.
So it can make. Then what happens on your skin?
Well, it doesn't. Nothing happens on your skin. It's actually a hole that they drill right into your subcutaneous tissue. So
They hatch?
Well, that's already hatched. This is the larva that's now going to grow up. And when it finishes growing up, in animals at least, it crawls out of that, falls onto the floor, pupates, and in a matter of days it exits from its pupa as an adult fly. So it's got a weird life cycle.
So people get these?
Yeah, they do. They
Routinely?
get botfly infections. In fact, if you went to Google Images, you can see some disgusting versions of them.
So you get kind of ulcerations on your skin?
No, there's not an ulceration. There's just a little black hole.
Oh.
And that black hole is the spiracles under which the insect is breathing.
Wow.
So one of the ways of trying to get this thing to crawl out of your skin is to cover it with Vaseline. So you'll seal off the air from the insect and get it to come out by itself. It has recurved teeth along its body, however, to hold it in place.
Interesting.
So it's very difficult. If you grab it with a forceps and pull, you'll likely just kill the larva rather than pull it out.
So is this a parasite?
It is a parasite, and it is under your skin, so it's not an ectoparasite in that sense. But
Interesting.
everything that infects skin, and I'll just go into that just briefly, like mites, for instance, scabies,
Yeah.
they live in the upper layers of your skin, almost the dead layers or the cornified layers of your skin. Is that an ectoparasite or is it an endoparasite? It's below the skin.
So it's an endo.
I would call it an endo. I would. So
But there's no negative consequence except maybe getting infected or something.
No, it's just a matter of location. So
It's weird, too.
It's more or less like us discussing whether a virus is alive or dead. Well, it's...
How does the botfly lay its eggs in the mosquito?
It catches it in mid-flight.
Are you serious?
I'm dead serious.
So the botfly is flying around.
Right.
It catches a mosquito.
Right.
And then
A female. It has to be a female
and
mosquito.
it lands?
No, it doesn't land. At the same time that it's flying around, it everts this mosquito and it lays its eggs with its ovipositor on the abdomen of the mosquito and then releases the mosquito.
Oh, my gosh. What countries does this happen in?
A lot of tropical and subtropical countries.
So
So a lot of people who come back from, like, Costa Rica,
I see.
and they went to the beach and they laid down on the beach.
Yeah.
They got bitten by mosquitoes.
Oh, my gosh.
They would have remembered a botfly. That damn thing is the size of a B-52 compared to a mosquito. And they come back, and then they're there for weeks, and all of a sudden they start seeing these lumps on their skin with little black holes in them. And underneath there, of course, is a larva about the size of a quarter.
So if you went to a dermatologist, what would they do?
Well,
they'd say,
of
oh,
course,
that's a
no.
botfly.
Yeah, but a dermatologist, of course, is paid to operate. So the dermatologist will put your skin under a local anesthetic and do a surgical removal.
Wow.
The most disgusting one we've ever shown in public was one that was removed from the lower eyelid of a small child.
Mm-hmm.
And to see this operation is absolutely remarkable. To see what that lump under the eye really turned out to be was an 1.5 inch long larva of a botfly.
Wow. So you've seen this here, Dickson?
Oh, yeah.
Wow.
Oh, yeah.
I guess in New York, with so many people coming back from exotic places.
And we have one of the premier dermatology departments in the world.
Here at Columbia?
Yep. It's fantastic.
Okay. Do you think that's good for Mike?
I do. I mean, we could go on, but I think we
All right, that's
probably
good.
shouldn't.
All right. We have two more. Okay.
Right.
I'll take one.
All right.
And you'll take two.
I will.
Next one's from Jim, who sends another story. Jim just sent us the story about the nematode in the guy's mouth.
Oh, yeah.
This one is, quote, a scientific discovery falls out of this epidemiologist's nose. Imagine finding a tick up your nose and being happy about it. Epidemiologist Tony Goldberg was working with primates in Kibale National Park in Uganda, but when he returned to his lab at the University of Wisconsin, he began feeling some pain in his nose. That's when Goldberg discovered a tick inside his right nostril.
Lord.
And he took it out with a forceps.
Right.
And then he analyzed the DNA of the tick, and it turned out to belong to the genus Ambyloma. And apparently it's a brand new species.
Nice.
We're still learning the pathways that diseases can use to move between wildlife and people, and this nose tick is a slightly amusing and particularly gross example of how diseases move in nature, Goldberg said.
Right. Amblyoma happens to be a soft tick, by the way, not a hard tick.
A tick in his nose. Well, that can happen, right?
Absolutely.
I mean, soft. What do you call it? Mucous membrane?
No,
right?
I don't mean that. The tick itself.
I know, but I'm saying that it would go fly up there, right? But it could.
They don't fly.
They don't fly. They hop up there. How
They
would it
don't
get
even
in?
hop.
How would it get in your
They
nose?
slowly crawl.
Crawl. So you brush into them on the brush. It starts crawling. Now, why would it crawl into this guy's nose?
It's rare. That's very rare.
I mean, just think. This guy has had to have this thing crawl up its face, maybe while he was sleeping or something.
Could be.
Weird.
Could be. All right.
Take the next one from Raihan.
Raihan writes, "Dear esteemed and honored professors, no wonder you wanted me to read this."
Not me.
I'll take credit for it.
I am neither esteemed nor honored. You can
writing-
take all of that, Dickson.
All right, here we go. I'm writing in response to TWiP 57, where in the intro, Professor Despommier mentioned that he had just returned from Singapore. The question was asked if water spins clockwise or anti-clockwise down the toilet, since Singapore lies 1 degree north of the equator, and I am pleased to announce that as a Singaporean, that water spins anti-clockwise in Singapore. I just did this experiment in my lab sink and toilet. Finally, I've produced some research worth noting. I don't want to disillusion you, but Rehan, we've already covered that subject and determined that the gravity situation is not strong enough for small bodies of water to induce the Coriolis effects. But
he
that's
says
okay.
he's in Singapore, and
He
it
did.
does.
No, he did. He says, okay, apparently the fact that it does not spin clockwise here has been noted before. Check out the last paragraph. But I'm still researching as to why this is so. I do know that the Coriolis effect is strongest away from the poles, and that it does not have much of an effect on small sinks, i.e. you can easily push water in the opposite direction, and the water would willingly change its course. But none of these explains why water rotates anti-clockwise. Rehan, a student in virology, not so much in the Coriolis effect. So... yeah, well, Vincent, you take it.
But we had a few letters before which said that the effect is not strong enough to influence the spinning of water.
That is correct.
But he links to an article in ABC Science, which is an Australian website, I guess, and which says that, according to popular myth, in the southern hemisphere, all cyclones spin clockwise, and so does the water in toilet bowls. But water going down the drain, half the time it's right, half the time it's wrong.
Right.
So it depends, and I don't understand it. And so Raihan is in Singapore, right? And he says he just went and looked.
How about that?
He did an experiment. He flushed his toilet.
You have a great city, by the way. Singapore is a wonderful place to visit.
All right, the last one is from Jim, who sends a link. His is third email to an Australian video,
Okay.
which you might like, Dickson. It's called The Most Horrifying Parasites. And it's a cute video. It is drawn in the way where these guys, they write on the screen, and you can see them writing, and they're just little cartoons.
We're going to play it now.
MarineParasites.com. There you go. Thank you, Jim. It's nice, right?
I love it.
It's hand-drawn, right?
I love it. There are some small tweaks that I would recommend to them. But for the most part, like you can't get pubic lice from wearing other people's underwear.
Okay.
Why is that?
They probably don't survive long enough. No?
Because they don't live in your underwear.
Pubic lice?
They live on your pubic hair.
Yeah, but don't they fall off into your underwear? No?
No way. They wouldn't dare do that.
They want to stay.
They want to stay. In fact, there are three kinds of lice, right? There are head lice,
Right.
body lice, and pubic lice. Head lice. How do you transmit head lice?
In schools, kids transmit it through their clothing.
Right. How do you transmit body lice?
I don't know.
Clothing. How do you transmit pubic lice?
Sex?
Got it.
And not by wearing someone else's underwear.
I'm sorry to say that, but that's true. So
All
all
right.
you transvestites out there, forget it. You can't
Transvestites? Why?
Yeah, because they will wear other people's underwear sometimes.
Oh, I get it. They don't have to be a transvestite. They could be a cross-dresser.
Cross-dresser. They could be a cross-dresser, too. They could.
All right. Now, this is not This Week in Transvestites.
No, it
Okay.
isn't. Or even in Transylvania, but we did discuss
Pennsylvania.
bats.
All right. That was a good TWiP, right?
I loved it.
Do you know where you can find this episode?
I had no idea.
Microbeworld.org/TWiP or iTunes. And we would love if you went to iTunes and subscribed. And if you like it, you could leave a few stars there or a few words. That helps to keep us prominent.
Yeah. Also builds up our damaged ecosystems.
Now, for some of this is a little technical now. For all of those out there listening via iTunes, you should not see any difference. But recently, I changed the RSS feed of this podcast.
Uh-oh.
And
Is
iTunes,
it a better feed now?
It's under our control.
Ah, that's
better.
even better. That's much better.
And iTunes will change immediately, automatically, no problem. But if you use other kinds of podcatchers like, Stitcher, I changed it with them. I don't know. There are a bunch of others out there that might not change automatically. So I'm going to tell you right now that you should change your podcast feed if you haven't been getting TWiP lately.
Uh-oh.
And to help you do that, I'm going to tell you what the podcast feed is. I will put it in the show notes.
Thank God.
It's twip.microbeworld.libsynpro.com/RSS. Okay, you know how to spell TWiP. You know how to spell Microbe World. Libsynpro is LIBSYNPRO, one word, .com/RSS. That is the RSS feed. If you don't know what I'm talking about, don't worry about it. But for those of you who have wondered where the show went, I changed the RSS feed. And as I said, not all podcatchers, which is a name for a program that will play your podcast.
But you can still get them on the internet just by typing out
you
TWiP.
could still get them on the internet.
That's easy.
You can go to microbeworld.org/TWiP and play it right there, or you can go to iTunes and subscribe. It still works. But I just wanted to let you know, in case 10 of you out there have a problem.
Right.
And, of course, do send us your questions and comments to TWiP at twiv.tv. Dickson Despommier can be found fishing. Also,
No, he can't be found fishing.
that's
Because
the problem.
when I'm fishing, I can't be found.
You don't see other fishermen or
I
fishing
try not
people?
to. I do it because I like to be by myself.
Do you ever see women fishing?
Rarely.
So it's a guy thing, huh?
It is, unfortunately.
I wouldn't like that.
Well, I don't go to see other people, Vince. I go to see the fish.
I see.
I go to see the fish.
Racaniello.org, medicalecology.com, and verticalfarm.com. You're going to start blogging somewhere?
Yeah.
Where?
Huffington.
The Huffington Post?
That's already been done. I have my first post up already.
It's set up?
Yeah, it's
Dickson, Despommier. Let me search for it. Huffington Post. Let me see if I can get it here.
I'm sure you can.
A bright future for urban farming.
Bingo.
Wow. It's the first hit when I searched for Dickson Despommier. Huffington Post.
How about that?
Posted out on the 21st of October. And the last 10 years have witnessed a rapid growth of urban agricultural initiatives in many countries throughout the world.
Okay.
You don't want me to read the whole thing?
I hope you don't read the whole
Are you getting paid for this?
Of course not.
Do you get a lot of hits?
I don't know.
There's some comments.
Good.
2 comments.
I'll go back and look.
Urban farming in Brooklyn has been brought to you by rural New York's water supply. And then another one says hydroponic food is bad, like GMOs. Boy.
That's too
Good
bad.
luck, Dickson, with HuffPo. But I'll put a link
Hydroponic
to
food is bad, like
HuffPo.
GMO. That's
Now,
a sign of
Dickson's-
extreme ignorance.
He's also at the Huffington Post. What is the name of your thing at the HuffPo? Do you know?
I just use my name.
I know, but do they... Oh,
It's
Dickson
under food.
Despommier, look.
It's under food.
And you can get updates from Dickson via RSS also. You can subscribe to his RSS feed. You can like him on Facebook, which I'm doing right now and makes 2 likes total.
Ooh.
See how much I help you, Dickson?
You do.
I'm Vincent Racaniello, and you can find me at virology.ws. The music that you hear on TWiP is composed and performed by Ronald Jenkins. You can find his work at ronaldjenkins.com. You've been listening to This Week in Parasitism. Thanks for joining us. We'll be back soon. Another TWiP is parasitic.